Hi, my name is Dana Raphael. I'm a senior in the college studying neuroscience and French, and today I'll be presenting to you a research project that I conducted this summer under the mentorship of Dr. Amelia Aish and Dr. Danielle Barber at the Children's Hospital of Philadelphia. So my research project is titled Microglial Cell Density and Morphology Classification in the Hippocampus of Early Postnatal Female and Male Mice. Now, just to give a little bit of background, microglia are the brain's resident immune cells. They play an important role in both the maintenance of normal brain function, for example, by clearing debris, pruning synapses, modulating neurogenesis, and even regulating cognition and mood-relevant behaviors. And they also play an important role in the brain's response to injury and disease. Now, the hippocampus is one brain region of focus for microglial study due to its critical role in mediating cognitive functions, including learning and memory, as well as its implications in mood regulation. And quite interestingly, microglia are sexually dimorphic cells. Previous studies have reported that microglia are distinct in male versus female rodents, even in early life. And specifically in the hippocampus, which continues developing well into the postnatal period, there are sex, postnatal day, and subregion-dependent differences with regards to microglial number, density, and morphology. That said, sex differences in hippocampal microglia have not yet been examined in the P10 mouse hippocampus, which is a very crucial time point given its approximate neuroanatomical alignment with human full-term gestation. Additionally, sex differences have not yet been assessed in key subregions of the hippocampus, including CA3 and the dentate gyrus hilus. Now, this is an image that represents a microglial cell. You can see in the middle, this is its soma or cell body, and here are its processes extending out and surveying the environment. Over here is an image that represents different microglial morphologies. So you can see ramified microglia as well as more amoeboid microglia. And this image over here is just to show that male and female microglia are not the same. So more specifically, the objectives of our specific study were twofold. First, we wanted to quantify microglial cell densities in 4 hippocampal subregions, which were the dentate gyrus molecular layer, the dentate gyrus hilus, CA1 and CA3, in P10 male and female C57 BL6J mice. And then we wanted to classify microglial cell morphologies in those 4 hippocampal subregions from the same P10 male and female mice. And our overarching goal with this project was to assess for baseline sex differences in both microglial cell density and morphology in the naive mouse hippocampus at the P10 time point. So that means that the tissues that we were using came from brains of uninjured mice. And now just to give a bit of an overview of the methods that we use to conduct our experiments. First, the brains were extracted from the mice on postnatal day 10. Then I sectioned the brains coronally on a microtome. We then mounted those coronal brain sections onto slides. And then we performed immunohistochemical staining for the microglial marker IBA1. And then those slides were imaged under the microscope. The microglial cells were quantified, the density was determined, and their morphology was classified according to 4 different categories, which I will soon touch upon. And this diagram over here just shows a little more in depth the process of immunohistochemistry, as I was very deeply involved in that aspect of the project. So over here, this represents a microscope slide with one brain section. We were staining for the antigen IBA1. Then we used a primary antibody against that antigen. So we used rabbit anti-IBA1, then a secondary antibody attached onto the primary antibody. An avidin biotin complex was used for amplification of the signal. Horseradish peroxidase was used to quench endogenous peroxidase activity, and diaminobenzidine was used to visualize the immunostained cells, and a fast red counter stain was also used. And then over here in figure 2, You can see the different subregions that we included in our analysis for cell density and morphology of microglia. So over here, labeled as 1, in this box, specifically this pink region, represents the dentate gyrus molecular layer. 2 represents the dentate gyrus hilus here in the middle. 3 represents the hippocampal CA3, and 4 represents hippocampal CA1. So those were the regions that we included in our analysis. And over here in figure 3, you can see the different morphological categories that were used to classify microglia. So we determined whether each microglia that we found in our photomicrographs, for example, in an image that looks like this, but a little more zoomed in, we would classify the microglia based on their morphologies. And it was our task to determine whether the microglia represented more of a round, stout, thick, or thin morphology based on previously published categories in the literature. And now to go to our results. These graphs illustrate our results. Over here on the left, we show that quantification of microglia cell densities in the 4 hippocampal subregions analyzed were not different between male and female mice at postnatal day 10. So that means that there was no sex difference in microglial cell densities between the male and female mice. However, there was a sex difference in microglial cell morphologies in these mice at the P10 time point, specifically in the dentate gyrus molecular layer. Female mice had significantly greater percentage of thick microglia in comparison to males, and males had a significantly greater percentage of thin microglia in comparison to females. In the dentate gyrus hilus, similar to the molecular layer, there was a significantly greater percentage of thick microglia cells in females versus males. In the CA3, there was a borderline significance of females having fewer of the thin microglia in comparison to males. And in CA1, there was not a significant difference between males and females. So just to conclude, while no sex difference was found in microglial density between male and female mice in the 4 hippocampal subregions at P10, our analyzed data does show a sex difference in microglial morphology in the molecular layer and hilus, as well as a borderline sex difference in the CA3. So more specifically, female mice had more thick microglial cells in the molecular layer and hilus, as well as fewer thin microglial cells in the molecular layer and CA3 in comparison to males. However, additional analyses are needed in order to evaluate the implications of morphological differences on microglial function, because currently the meanings of these morphological categories are still highly debated. So for example, in the adult rodent, similar microglia morphology categories as those that we used, they are considered to be a gradient reflecting the degree of activation of microglia. So round microglia, such as that represented here in image A, they're considered to be more active than the ramified and presumably surveying microglia, which are thin in this image D. But in contrast to that, in the early postnatal rodent, which aligns more closely with the time points that we were using in our study, microglia morphological categories were proposed to reflect the degree of age or maturation of the microglia. So for example, these round microglia are considered to be younger or less mature than the ramified thin microglia. So given all of this, with our analysis, it's not exactly possible to know whether the greater percentage of thick and lower percentage of thin microglial cells in the female versus male molecular layer means that microglia in female mice are younger or more active than those in male mice. So additional analyses are needed to evaluate for that. And just to touch a bit upon our ongoing and future directions, we will be quantifying microglial cell densities and classifying their morphologies in P10 intermediate and posterior hippocampal sections, because although those have been stained during the summer, only the anterior hippocampal sections were included in our current analyses. And in addition to that, we will also be classifying microglial densities and morphologies at additional time points such as postnatal day 3 and postnatal day 7 in order to compare the developmental trajectory of microglia between male and female mice. And in addition to that, all of these studies served as a baseline that will be crucial in further studies that we will be conducting in the lab to assess whether there are sex differences in microglial cell density and morphology following injury. Specifically, we will be using a mouse model of perinatal hypoxic ischemic brain injury to model the human condition of hypoxic ischemic encephalopathy, or HIE. And this schematic over here illustrates the methods that we will use in those experiments. So each mouse will receive either ischemia or sham surgeries by having a carotid artery ligation. Then those mice will undergo either hypoxia or normoxia, and then we will be able to analyze the microglial cell response as well as different behavioral assessments. And lastly, I would like to acknowledge several individuals for supporting this research project. Specifically, I was supported by the Neuroscience Program Fellowship for Summer Research, sponsored by Penn's Neuroscience Department. And I was also supported by the Mary L. and Matthew S. Santarocco College Alumni Society undergraduate research grant, sponsored by Penn's Center for Undergraduate Research and Fellowships, or CURF. And I'd also like to thank the Aish Lab members, and especially my mentors, Dr. Amelia Aish and Dr. Danielle Barber, for their unwavering mentorship, feedback, and support throughout this project. Thank you so much for listening to my presentation of my research project, and please feel free to contact me with any questions. I'd be more than happy to answer them. Thank you.